Hello, everyone, and welcome to Gene-Based Therapy in Action, behind-the-scenes look at genetic therapy trials. My name is Katie Howe, and I am the Clinical Trial Navigator for the CF Foundation. I will be helping with today's presentation and introducing our speakers in just a moment. But before we get started, I want to go over some logistics and reminders. This session is being recorded and will be available after the event on the CF Foundation YouTube channel. You will receive an email when session recordings are available. We will be taking audience questions during the last 20 minutes of the session. You can submit your questions at any time during the presentation via the Q&A tab. Although we cannot answer questions about individual care and treatment plans, we encourage you to use the information you learned today to have conversations with your care team. We will do our best to answer as many questions as possible today, including those that were pre-submitted with registration. Before we begin, I want to remind everyone of our event conduct guidelines, linked in the chat. Please be considerate, open-minded, and respectful. We ask that you do not use malicious or offensive speech and do not ask for or give medical advice. If at any time you feel the event conduct guidelines are not being followed, please contact a CF Foundation staff member at the help desk. Finally, if you experience any technology problems during the session, please visit the help desk in the expo area on the left-hand side of your screen. Now let's get started. I'd like to introduce our panelist speakers. Samantha Sam Johnson is a senior clinical research coordinator at National Jewish Health in Denver. She joined the team in June of 2022 after earning her Bachelor of Science in Neuroscience from the University of Minnesota. Over the past 3 years at National Jewish, she has played a key role in recruiting participants and conducting study visits for several nucleic acid-based therapy trials. Samantha is now preparing to attend medical school this fall at the University of Washington School of Medicine. Dr. Ahmet Uluer is the director of the Adult Cystic Fibrosis Program and co-center director of the combined Boston Children's Hospital and Brigham and Women's Hospital Cystic Fibrosis Center. Dr. Uluer is also a general med-peds trained pulmonologist who cares for both children and adults. He is the director of the Therapeutic Development Network locally and was recently the North American lead of 2 phase 2 clinical trials. Steph Hansen is an adult living with cystic fibrosis from San Antonio, Texas. Steph is passionate about bringing awareness to patients who come from diverse backgrounds, as well as those like her who cannot yet benefit from CFTR modulator therapies. She currently serves as a member of the CF Foundation Adult Advisory Council, as well as a reviewer for community support grants. Now, I'd like to turn it over to Steph to tell us how her experience with genetic therapies all started. Steph?
Good evening, and thank you, Katie. I appreciate the introduction, and I'm so grateful to be here with all of you this evening who are tuning in all over. I'm Steph Hansen. Like Katie said, I'm 35, living with CF that's caused by 2 nonsense mutations. And in the last couple of years, I had the awesome experience of participating in a clinical trial. After the release of Trikafta, I had a couple really tough years, and I ended up feeling really lost and forgotten and left behind by the CF community. And it eventually occurred to me that taking action through participating in research or clinical trials seemed like it might help combat some of those feelings of me feeling left behind if I was actually doing something about it. And what first piqued my interest around genetic therapies was their potential to treat all people with CF, no matter which mutations caused somebody's CF. And I wasn't seriously looking, but in February of 2023, I got an email from Emily's Entourage from their registry that led me to connect with a team in Boston. And it was only their team in Boston was only one of 2 sites open at the time and enrolling in this clinical trial. So I got connected with their team, and I was really unfamiliar with different genetic therapy trials, but Dr. Euler's team was actually the team that I ended up working with. And once I got in touch with them, they did a great job of sort of educating me about what it was that I was signing up for. I was really uneducated, didn't really know what I was getting myself into as far as genetic therapies or clinical trials go at all. This is my first jump into um, clinical trials and research at all. So while I've got a solid grasp on it now, this is not a topic that anybody wants me to try and educate them on. So I'd like to turn it over to Dr. Euler for a bit so that he can talk to us about different genetic therapy trials that somebody might encounter when looking into research opportunities. So Dr. Euler, can you explain to us a bit more about these different types of genetic therapies and the different vectors that people might encounter when searching for trials to participate in?
Absolutely. And Steph and Sam, it's such an honor to be here together with you guys. And Steph, so grateful that you reached out to us and that we had an opportunity to, work together. So I'm going to get the slides up here. So I'm going to speak with some slides, but this is really going to be hopefully a discussion that we'll be having, like just going back and forth. These are my disclosures. And essentially, we need to go just back to the beginning slightly, just talk about genetics before we can talk about some of these therapies. So just to give some people background, the majority of our genetic information is stored in 23 chromosomes, which you might remember from biology class in high school. And each cell contains the same information, though not all the cells, use all that information. And so the blueprint or the product includes, so essentially each chromosome contains DNA. That's the blueprint or the instruction manual of our body. And that blueprint also includes the products that are going to come from it, our proteins, and all that information that we need to grow and function and even reproduce. So we're going to start blowing up this cell and to look at its contents, and we're getting closer to those chromosomes. And inside those chromosomes, we're going to see the DNA. Essentially, and I hope I don't regret this analogy, but I'm going to use a cookbook analogy. And this is a picture of a cookbook I found in my kitchen that aptly says everything you need to know in the cookbook, know how to cook, and like our DNA. And so, sorry, just coordinating this. So anyway, our DNA helps make up some of these unique characteristics, and also it is important to know that our DNA is made up of genes. Now, these genes are what makes us make up our unique characteristics, and a gene is like a recipe in a cookbook. It talks, tells our cells how to make a specific protein, like insulin, like hemoglobin or collagen. And for our discussion, it's going to be the gene that causes cystic fibrosis. So the gene, or in this case, cystic fibrosis transmembrane conductance regulator gene, or CFTR from now on, is one such gene, and if altered, results in cystic fibrosis. And genes made up of DNA provide the instructions to make RNA, which is a temporary photocopy of the DNA that, I'm just realizing that I had some animation to go with these slides. That animation doesn't happen here. But essentially, they're photocopies or temporary photocopies of the DNA that they don't last very long, but they make up our protein. And mRNA is like that temporary image, and it's the next step in that recipe to make that protein. And in this case, CFTR, which is coordinating the movement of chloride and bicarbonate, and those are the important functions of that protein that are going to allow us and our lungs or any of the tubes inside our body, to function appropriately with their secretions and movement of chloride, with the movement of chloride, and also controlling pH, or the acid-base balance. And so in this case, this protein is analogous to the main dish, or CFTR is the main dish that comes from that recipe. So in order to sort of just, go through that full information, and you would have seen first the top line here, but essentially it was the DNA that's transcribed into RNA, which is then translated into a functioning protein that helps, the other architects in the cells, things like called ribosomes, make this functioning protein. But bringing in our analogy, you have the DNA cookbook that contains the genes or the recipes that are then copied to that temporary recipe, so that you don't have to worry about that recipe. You can get make spaghetti sauce on it, you can like burn it, you can ruin it because it's temporary and gets removed by the cell and degraded. But then the chefs use that recipe and put it all together to make that main dish or the protein. And you can think of mutations as essentially where the ingredients of that recipe have gone wrong. You've gotten some substitutions, and sometimes those substitutions lead to a protein or a product or a main dish that tastes okay, and maybe there's still some CFTR function left. Or there's enough CFTR function. It doesn't, taste like the main dish, but it is a protein that can be restored if possible. Or sometimes you can it lead to a recipe that just does not work and nothing is created. I'm going to actually skip these next two slides, but they'll be included in the slides here that if you're interested in how that mutation is inherited, because I'm not sure we need it for this part of the discussion. But essentially, now that we're managing CF and our research is always evolving, developing therapeutics that are directed towards symptoms, and we're still working on these strategies as our previous panel, speakers have spoke on, like better pancreatic enzymes, better antibiotics, better nutrition. But we're now evolving, and we have evolved into the next phase of therapeutics, and the standard of practice now is small molecule therapies directed at rescuing the CFTR protein. These are called modulators, as you know, and they've changed the lives of so many. And while they help close to 90% of people with CF, it leaves behind approximately 10% like Steph, who are ineligible or experience side effects. And it's this approximately 10% of people who we're counting on to come up with, who are counting on us to come up with these alternative therapies that will transform the lives like those who are on modulators, and how that has happened thus far. So this next phase is where gene-based therapies comes into play, and we're utilizing DNA and RNA and genetic material like oligonucleotides to get to that next phase of therapeutics. And again, we know about, to the left, you're going to know about the CFTR modulators, but focusing on now restoring CFTR genes or fixing or replacing that incorrect gene. So in the graphic would have shown the slide open up to the left, where restoring can happen by using small molecules, and we're going to and that skip an error part of the DNA or the blueprint called read-through therapies, and we're going to skip that for now. And our focus is going to be on mRNA, which works by introducing a copy of that correct recipe from the cookbook to cells so that they can make the correct protein or that main dish. There are also short bits of genetic code that can be inhaled, and for certain mutations, like splicing mutations, and can act as a tiny patch over that error in the part of the genetic code and help fix that CFTR protein being made at the RNA level where it is needed the most. And then gene transfer refers to the DNA transfer as a way to give the body a working copy of the gene, like providing a corrected recipe to a damaged gene within a cookbook. So that mutation is already there. We can talk about this a little bit later, but we can utilize things like viruses that are rendered harmless to act like a delivery truck of corrected genetic material, and delivers the ingredients for a corrected recipe to that cell. And gene editing is where we fix the recipe still inside that cookbook before it becomes RNA and the protein. So gene editing can be done with cells inside the body or with cells that have been taken out, corrected in a lab, and then put back in the body. And similar to the curative treatments we can see in sickle cell disease, or for those who are familiar with it. And it's unfortunately a more complicated pathway for treating CF, but we're already seeing so much of science fiction come to life and reality everywhere we look these days. So we know that those days are not too far behind, we hope. The gene editing can be done in a number of different ways, and that's going to, we see that in the far right, whether it's through CRISPR, Cas9, as you know about, and the part of Nobel Prize award, and zinc finger nucleases is another name, or transcription activator like effector nucleases. Anyway, these are names of different techniques to edit the genes, but we're not quite there yet, and those are coming. But we're going to focus on therapies that introduce a corrected copy of the blueprint called gene replacement therapy. I think that this popped up, right? Gene replacement, or also gene transfer or gene addition. And this involves introducing that healthy copy, as we talked about, of CFTR into the cells for people with CF. And typically we use a vector to deliver this to the healthy gene, to the cell, and these will hopefully take over the function of that defective gene. And as you can see here, there are, before I turn it back over to Steph, and Sam, these are clinical trials that are ongoing right now for people that are living with CF in the US and also in Europe and elsewhere. The first one is by a company called SpySense, and they have that little strand of genetic material that you inhale without a vector, actually, that gets into the cell, hopefully, and will patch up that incorrect part of the mutation and help things work. To then the companies that are like Vertex and Arcturus and Recode, who are using lipid nanoparticles, and we'll talk about this later, as a way of ferrying in mRNA, or in this case, mRNA into the cells. And then there's also the viral vectors that are being used. And these are companies like 4D, Spiravant, Crystal Bio, and then also Boehringer Ingelheim using a different type of virus that we'll also talk about. So hopefully that'll give you just a brief window into so what we're discussing here today. Steph?
Thanks for going over all of that, Dr. Uluer. I remember when I first got connected with your team, I was just so excited about the possibility of doing any kind of trial that could potentially treat any mutation, anybody with any mutation that causes their CF. I was just like, just sign me up. Like, I don't even care. Whatever, I'm ready to do it. But, we had to do our due diligence. And, once I understood more about the trial, I remember we had to make sure that, I was a good fit. And based on the initial inclusion and exclusion criteria. And first things first, I remember I think within the first week or two, I got connected with your research coordinator, and she became my main point of contact from that point on throughout the rest of the study. And I worked with the research coordinator to start the process of getting my medical records sent over. And while there was a whole lot of waiting around on my end, I know that there was a whole lot happening behind the scenes to sort of prepare for my initial screening visit. And so I was wondering, Sam, if you could talk to us about a screening visit. What is a screening visit? And as a research coordinator, or RC, what kind of work goes into preparing for a screening visit?
Yeah, I'd be happy to. Thank you, everybody, for being here and listening to everything we've put together today. Also to Dr. Uluer and Steph for putting together such a great panel. A screening visit is research way of saying a baseline visit. It's the way that a research study kind of assesses where the person with CF is at. So you'll do a lot of things that are usual to you: spirometry, blood work, ECG. You'll meet with a doctor. You'll talk about what medications you're on. And then for these trials, a lot of the times, we'll have some additional pulmonary function tests, sometimes some imaging that we do. And then we go through all of your medical history and kind of get to know you when you're in person with us. And it is a small, shorter visit to the long process of getting approved for a screening spot. And that's kind of what happens over those 3 months that you are waiting, is that we are getting medical records and kind of trying to learn your medical history with CF and kind of what's happened to you and if you'll qualify for this study. And then usually we do have to get a spot from the sponsor and a date approved. And with these trials, that can be some of the hardest timing because of how they have to have certain windows line up and with everybody's schedule, it can feel like trying to find a unicorn, is how we always refer to it, of days. Of the doctors here and radiology is available and the RC and the sponsor is going to let us do that as well. It's a lot of back and forth over that time, and it is 3 months and it's a lot of waiting, but it usually goes by pretty fast, I say.
Yeah, you're totally right. It did go by really fast, but at the same time, I feel like I was so anxious to just get things going. I was ready to get to Boston and start dosing and all of those things. So I was really excited, but it sounds like it's a whole lot of work for you research coordinators. You guys are like magicians, I feel like, because I got to just hang back and, all I had to do was get on the plane and get to my screening visit. But you guys have so much to coordinate and think about. And, it's a lot on us, I think, as patients too, because like you said, there's, I had to keep my whole life kind of on pause, for a period of time there to make sure that when the days lined up for dosing and screening and all of that, that I was ready to go. So yeah, it's a lot to coordinate. Coordinator is part of your title is like definitely accurate. It's you guys are magicians though. So, but I arrived in Boston for my screening visit, and I realized really, really quickly that research is a totally different arena than routine CF care, which is a place where I know which questions to ask and what my needs are and stuff like that. But as I went through the informed consent with the RC that I was working with, I was really surprised to find out at screening that, this particular trial would require me to get on birth control, or there were like a few other things that popped up as we went through the informed consent. And so, I realized that I should really be asking more questions. A lot of those things were caught because the research coordinator was going through all these things with me, but it just made me realize uh, how many questions that like were going to come up, and I shouldn't be afraid to ask them. But that screening visit was it was also my first time meeting Dr. Euler, and although I was interested in a particular type of trial, I got to ask him about some of the other sort of thoughts and concerns I had. And so during this screening visit, Dr. Euler, I got to talk to you about how I felt a little bit apprehensive about some of the different methods of administering these drugs. And, could you talk to us about the safety of these trials and how they're set up and stuff like that?
Yeah, I absolutely. And I'll just add to that. I think I mentioned this before, but I mean, people like Sam, I mean, definitely to do that job, you have to have like executive function skills extraordinaire. And that thing that you just mentioned, when things line up perfectly, there's nothing that makes our RC like so happy, like just that everything on the calendar lines up, we got everything covered. So then if something like, impacts that, boy, then the disappointment is just as as bad. So like very quickly, I mean, things that you want to know about when you're, like agreeing to take on a trial is, how are things delivered? Where are they delivered? What cells are being impacted? Things that you may not know to ask about. So, like for example, modulators affect the whole body. So we see how like a systemic treatment might be beneficial. And in this case, as you saw with all the clinical trials on that slide, that we're inhaling those things, so they may get to the lung and also partially to the sinuses, and so that's easy to deliver to the cells. And of course, there's pros and cons to each of these methods, but we're if we're successful getting these cells to take up the genetic material, it will unfortunately involve only the cells in the lungs and the sinuses. So the rest of the body won't get it. But this is a good place for us to start. And then also there are different types of cells that we're targeting, and these are like not things that I want you to remember, but it's things that we think about when we're designing gene therapies, or we're thinking about how we're going to, talk about this. We talk about where it's going to be targeted. We talk about the cells, and somatic cells are the type of cells that we are targeting right now. And they're not reproductive cells. They provide a temporary fix because those cells don't, survive, our entire lives. And so the longer benefit, depending on which cells take up the DNA, essentially is the case there. So if we get to the cells at the bottom of the epithelial layer called these basal cells, things might last a little bit longer because they're the cells that then lead to the other ones, and they're the type of early cell, not quite a stem cell, but like stem cells. They lead to and renew the other cells in that organ, and those can last a bit longer. And so next slide. So essentially, this is a complex soup of things that need to happen. And so, to very quickly sort of steer you through this, I'm going to show you this slide that essentially will, once again, not be animated. I apologize about that. But essentially, I'm going to go through this cookbook, analogy again, and hopefully it is working throughout this talk. But it is, efficiency in delivering is a challenge, and so we have to overcome some of those challenges. And so the delivery vehicle is what we want to look at, and that can be either a vehicle that has a viral capsule or protein code of a virus that allows the DNA or that cargo to be brought in. So that's like an infectious part of the genome is removed. And in like this one called an adeno-associated virus vector, we take advantage of that virus that has developed over thousands of years and of evolving. And essentially we've taken away the infectious part, but we've held onto the part that is good at getting into the cell. And so then it'll take that cargo, in this case, I liken the delivery vehicle as a sous chef, helping that chef get those ingredients, and those ingredients and what we need in the recipes are part of that cargo. And the other one, that non-viral vehicle we can see on the left side of the slide, those are fat molecules, lipoprotein nanoparticles, the same type of particles that are surrounding the RNA vaccines of COVID. Now they're a little bit less efficient than viral vectors getting into the cell, but we can give more of it. We can give it in a way that doesn't, as a viral vector, maybe cause an immune response, and so we can give it more and more, and then the genetic material, as you can see there, in that inside that cargo. So this one is just looking at a closer look at the lipoprotein that's coming in. Essentially here you can see the mRNA in that lipoprotein particle coming into the cell, and then it gets released into cytoplasm, where that's where the mechanisms are, and the chefs and other ones working on to make that protein, and hopefully in this case the one with the corrected recipe. I think it should say that there. And so then in the, looking at the next slide, which I'm going to just back up for a second here, we're looking at what we just talked about, the adenoviral vector, that is adeno-associated viral vector. And then we're also looking at another one called a lentivirus, and there's, different, benefits to those, to those cells. So let me just move this forward here. And in this case, both of those, ways of getting to the cell, they're able to use receptors on the cell surface, use other things that are that make it able to get into nucleus of the cell. And in the case of the adeno-associated virus, it's a non-integrating, meaning that it leaves its DNA within the nucleus itself that doesn't actually integrate with the DNA of the host, or DNA of our DNA. So it can, utilize the systems of the cell to make more of itself, but it doesn't necessarily get passed on when a cell divides or replicates. But when you have an integrating one, which is the lentivirus, and that is obviously a positive, because then you don't have to keep dosing or not dosing as much, that gets integrated, but then that comes with other potential concerns that we're hopefully mitigating as we go forward. And in this next slide, you'll see here that each pro and con is that in the mRNA one, you may need daily dosing. There may be some flu-like reactions to to that lipoprotein particle, and the innate immune system, the immune system that is reacting to a foreign object that comes into the cell. And so in these trials, however, and this is part of the question for Steph, is that how does it impact you being in other trials? What is the follow-up? Because when you're doing follow-ups often, then you can't really participate in other clinical trials. And this is true for some and not true for others. And then we look at the non-integrating and integrating viral vectors. We know that you are going to give less of them, less often. We are concerned about immune response, so that the follow-up, might be a little bit longer. And then, when we talk about safety, we know that there's modifications, things that have happened to these viruses that are hopefully addressing some of that immune response that we're hoping to avoid. And then just that last one, the nucleotide, oligonucleotide therapy, that can also cause flu-like symptoms as it tries to, get into cell and do its thing. And I think then we can end it there and then go back to Steph. And then hopefully that gave you, some of the information that you needed to decide on whether you want to do that trial. For you, it was that you're already there, too late. You're going to be a part of that trial, but gives you the more information, moving forward.
Well, I remember being at that screening visit, and the research coordinator was like, you can stop, you can, opt out of this at any time. And so that was like one of the main things that she said, and I'll never forget it.
And what I just said, saying too late, that was absolutely not true. It's never too late. You can always, and that's really important actually, is that you can always stop being in a clinical trial. You can always opt out at any point. All right, go ahead, Steph. Sorry.
Yeah, very true. But no, I was really grateful that you explained all of that. I think that, to understand as a patient how these trials are being designed and how our safety is being considered, it really empowers us to make the right decisions for ourselves and to know which clinical trial opportunities we want to take advantage of and opt into. And so, I felt really great after having that conversation with you at screening. So I did the whole screening visit, decided this is something I want to do. I'm not opting out. And so I went home and waited on screening results. And there was a whole lot of waiting during that period of time. I put a whole lot of my life on hold, kind of like what Sam was talking about earlier. This is like the beginning of summer. So I had friends and family reaching out to me asking like, oh, do you want to take this trip? Do you want to make plans and do this thing? And I was like, I don't know if I can, because I may or may not be opting into a clinical trial halfway across the country, so it was just a lot of hurry up and wait. But we were waiting on getting my genetics back. I think that took like 30 days. And it was so crazy how the timing of it all worked out, because my genetics took so long to get back. I was literally about to fly out on a Monday morning. And I got a phone call at 6:30 a.m. from the research coordinator saying, hey, everything's good to go. Your genetics are good. Like, get on the plane. And my flight was leaving at 11:30 a.m. that morning. So like just a few hours is all we had to confirm that everything was lining up. But as life with CF goes, I actually had to break it to the research coordinator on the phone that I was having a bit of a medical emergency. So I had to let her know, and unfortunately that meant that I couldn't fly to Boston that day, and we had to cancel everything last minute. So my health emergency forced us all to call off the dosing visit. And I really relied very heavily that day as I was driving to the ER. My RC helped me cancel the flights, the hotels, She really took everything off of my plate so that I could focus solely on taking care of myself. And I'll never forget, I felt so embarrassed and unsure whether the team in Boston would want to continue to work with me. But, our health is our health, and we've got to take care of ourselves. So um, I was wondering, Sam, this obviously happened to me. And I'm just curious, like, have you ever encountered something like this happening? How are emergencies or health issues handled within research, and how do you handle that with your team and the sponsor? Like, how often does stuff like this happen?
Yeah, of course. And just like you said, Steph, it is life with CF. All of us that work on these studies and work at these centers understand that we've asked you to be stable at a baseline for now probably upwards of 4, 5, 6 months. And that is just maybe not reality. And it happens. I'm sure every research team has a story. I know our team in Denver have stories of people the weekend before, the day before, having something happen. And not everything has to completely push off all of the schedule. There are things, flights get canceled, doctor's appointments get rescheduled, family members might not be able to come out on a certain day. That only push us back a few days. And then there are things that maybe we have to redo the whole schedule for a few months out, and that's all okay. As long as the research participant, as long as Steph's ready to go, I know every team will then be ready to go when you're ready again, when you're healthy, and when you feel back to normal. And the good thing about these trials is those 28 days, 30 days that we're waiting for all the results, we are in near constant communication with everybody involved in the teams at our center, the research participant, the research participant's family, the sponsor of the trial. So it's one email. We're all on the same email. It's one or two emails with the information that we know, and it is then handled on their end. It is our job to handle making sure the things are canceled. So I'm glad that was your experience, that we were able to, that the research coordinator was able to get all of that canceled and moved around for you, because just as easily as we schedule them, things can be moved. And everyone in hospitals is aware of how scheduling can be and moving things around. And I'd rather you be at home at a home hospital than fly here and then tell us, and then now you're in a hospital in another part of the country.
Yeah, for sure. That, I will just never forget being so in my head about it and like, oh my gosh, this, I must be the exception here, of course. But one of the things that my research coordinator told me on the front end was like, one of the hardest parts about research with people with CF is getting you guys to stay stable for an extended period of time, So, I'll always remember that when participating in clinical trials from now on. And it's so good working with teams like you guys, that, you're familiar with how the beast of CF and how it works and how it can sort of rear its head sometimes at the most unexpected or inconvenient time, But it helps having a great team of people ready to help however they're able to. So, in my case, I had to reschedule my screening visit several months later. I ended up in the hospital for like a month and needed surgery and all of these things, and then needed to stay stable for a period of time after hospitalization. So I finally made it back to Boston for a second screening visit. And this was 6 months after the first email to Boston. So like we're 6 months along now. It's been a while, but I finally make it back for my second screening visit, and I dose very quickly after that second screening. It was like a week afterwards. Dosing day, it came really fast, and I knew that it would be intense, but I was really impressed and surprised with how tight of a schedule we were on during dosing day. I have never in my life, and Dr. Euler said this earlier, but I've never seen somebody as organized as a research coordinator on dosing day. It was crazy that they had everything like laid out and labeled, that this blood sample needed to be taken at this time and all this stuff. It was, there was a whole binder that like had everything listed. So I'm curious, what does dosing day look like? Like, what do you have to do to prepare? And what does that look like from the research coordinator's perspective?
Yeah, it is just like Dr. Euler said, a lot of executive functioning that goes into getting to that point. We love our label makers and we love our charts. And I don't think any research coordinator would get through a visit like that without any of that. I know one of the good things in the waiting between screening and dosing days, it gives us as the research team time to kind of go through those fine details and kind of make a schedule of, so if we're hoping to dose at 10, then the person needs to come in at 8, and then we'll do this and have our schedule up until this point. And then our doctor can come over for the dosing, and it'll take about this much time. With all of that being said, it is a lot of teamwork, to the point of you'll meet more people probably on dosing day from the research team than you'll see for the rest of the study, because we've timed out, oh, we're hoping to do this blood draw here, and then you'll grab it and you'll take it and process it. So it's a huge team effort. Everybody's on board for that dosing day. We're all really excited for you, for the research participant, who's been ready to be at our site for however many hours. And we're also ready to test out all of the things that we've been practicing up to the day to get ready for it.
Yeah, you're totally right. I remember during dosing day, I was admitted to the hospital the day before, and I was in this hospital room that was it was one of those isolation rooms. So there's this sort of antechamber room between my room and the hallway. And at any given moment, I look into the antechamber, and there's like two like research interns in there, just like waiting around, ready to be an extra set of hands. And it's funny because I make a joke that like I felt kind of like a potato all day during dosing day, while the RC just really led the show. The team was there, and they were doing all the things that they needed to be doing at any given moment. And I was just like, okay, well, here's my arm for some blood, or here, let me do a PFT. And there were times of the day where we were literally watching the second hand on the clock to get things done at very particular times. It was intense, but I kind of just got to sit back and do whatever they told me. But then came dosing, and in my case, I was getting one dose. And so it was at that point Dr. Euler came in, and I'll never forget, he came in, it was for like an hour or so, and I was really anxious and excited to dose. And he was like, all right, this is probably going to be pretty anticlimactic. And sure enough, he was right. We got to dose, and him and I just chatted, and it was a little bit anticlimactic. But there was lots of follow-up testing and monitoring after dosing, and I discharged the day after, and my RC worked to help me not have to travel back to Texas, where I live, immediately after dosing. So I got to stay in Boston. I think it was like 10 days for my first couple of follow-up appointments after dosing. And I was blinded to the results of this particular study, which I thought was really nice. I was curious to see what the results were, but in the end, I didn't end up obsessing over the numbers or how exactly I felt after getting the drug. And I got to just kind of sit back and do my best on PFTs and stuff like that, and enjoy the experience of getting to participate in a really cool and exciting clinical trial. So that was really, I ended up enjoying that sort of part of the clinical trial, and not worrying so much about whether the study drug was working or not. But during that visit, we planned out the next couple of visits where I would come back to Boston and follow up with the team and be monitored and stuff after dosing. And we ran into several issues about the hotel being sold out, or whether it was going to be my partner accompanying me to Boston, or a friend, or would I have to make this trip alone? And it was great that my RC helped to quickly navigate any of those roadblocks that we encountered. And it seemed like once we were done dosing, scheduling for the follow-up visits became a little bit easier and more flexible. Sam, is that pretty typical, that scheduling becomes more flexible as you get further along in these studies?
Yeah, it's one of those reassuring pieces that we do ask for a lot of your time to be on hold at that beginning time, that first, 6 months leading up to a screening and dosing, and the first few visits after have to be pretty strict, even watching the seconds on a clock kind of strict. And then as we get farther out, a few months, of weeks, months post dosing, they give us a little bit more wiggle room. So we may have 2 weeks that we can pick a day for the visit, or 3 weeks to pick a day, instead of like it has to be this day or the day before, which is nicer for people because it kind of lets you be a person and have a regular schedule outside of research. But I would say it's pretty typical over most of these studies.
Yeah, like I said earlier, I think that towards the beginning, especially around screening and dosing, I had to tell my friends and family, like, I can't plan anything. I don't know what I'm going to be doing over the next couple of months. But once we got dosing done, it was much easier to sort of plan things out. Like, oh, maybe it would be cool to spend Thanksgiving in Boston, or, I got plans for Christmas, with my family, so maybe I can come in a couple days after Christmas or whatever. So that was much nicer and easier as things went along to be able to plan and to have some flexibility. But I think that I went back to Boston a few more times. It was like 4 or 5 more times, and over the next 6 or 7 months, and I was really sad to kind of see it all end. I was sad to not get to work with the team that I was working with in Boston, and I hope that I get to work with them again eventually. But overall, I had a lot of fun participating in this trial, and I learned a lot about research. I learned that it's similar to my normal CF care in regards to advocating for myself. When I first got in touch with the team at Boston, somebody mentioned to me, like, this is not going to be a vacation to Boston. Like, you're here for research. I was like, okay, yes, like, no problem. But I learned throughout my time and working with a really cool RC that I got to communicate my needs, and, they're always worth asking for whatever it is that I need or what I want. And the RC acted as sort of my advocate between me and the sponsor. And it helped to sort of have somebody in my corner advocating for me throughout this whole process. And so I ended up having a whole lot of fun throughout my time in this trial. I got to see a lot of Boston and eat lots of lobster rolls, which was really fun. If anybody ever needs a recommendation for best lobster roll in Boston, let me know. But there were also things that I learned that I should have probably been doing, but I didn't really know at the time, like asking questions. I mentioned earlier that this is a very different arena than my normal CF care. Research is very different. And so, like Dr. Uluer mentioned earlier, I didn't realize how my participation in this study might preclude me or restrict me from participating in future genetic therapy trials. So, I didn't realize that until afterwards, when now I'm looking to participate in more clinical trials and I'm not able to for a certain period of time. So, those are all things to consider. And I encourage, again, anybody wanting to participate in these kinds of trials to ask as many questions as you can whenever they occur to you. But as I suspected, participating in research, it really helped me to feel less alone and less powerless over being in that 10% of people with CF who can't take modulators. I felt like I was part of something much bigger than just myself. And it helped me to feel like I was making a small difference where I could. And, you know, there are so few people who can participate in some of these trials that, you know, it was up to me to step up when and where I could, if I could just stay healthy enough and stable enough. So, you know, it also, I think, expanded the resources that I have at my fingertips for me to stay informed on clinical trial opportunities. So, you know, I check in with the team over in Boston as often as I can to see if they've got anything new for me. And I'm really grateful to have had this opportunity. And I encourage anybody to reach out and inquire about any of these genetic therapy trials if you're interested and you think you might be eligible.
I was just going to say, what a powerful statement that you said about not feeling alone. And I would say also that this is exactly the type of thing that you were watching. You know, like people here are experiencing this, and then you made outreach because you wanted to participate, you want to do something. I'm hoping that this is exactly what ResearchCon is about, and the CF Foundation wanting to, you know, get people to want to participate, just like Emily's Entourage and just like, you know, when other organizations, do similar things like CFRI. So, like this is a great place for to be doing that here at ResearchCon. Oops, sorry about that. But I was also going to just say that one other thing I would just add that Sam, you talked about practicing. I think that that's really, you know, that's what you're seeing, Steph, but that practicing, can you just explain just a little bit more about that, maybe in just 1 minute? Like just like you do like a simulation.
Yeah, a lot of these trials I feel like almost have encouraged, if not required us to some point to get the team together and almost truly time how long it takes with our pharmacist prepping it, where are they at? Let's time how long it takes her to walk to you guys. And then kind of going back in between each of our time and doing handoffs in between each research coordinator and lab person. It's very much so like a dress rehearsal of the dosing day.
Yeah, and Katie just put on the chat, just the CF Foundation Clinical Trial Navigator is a great way of finding those trials. So thank you for posting that, and thanks to the CF Foundation for doing that.
Yeah, for anybody who hasn't been able to connect with Katie Howe, the CFF's clinical trial navigator, she's a great, incredible resource. You know, if you have any sort of idea of the type of clinical trial you want to be part of, or if not, you know, she has so much information. You know, you could tell her, oh, I'm interested in genetic therapies or phage trials or antibiotic trials or whatever it may be. And she is such an incredible resource to sort of give you tailored information for whatever it is you might be looking for. And, you know, for me personally, there's, you know, I'm not in a place that does a ton, like geographically, I'm not in a place that does a ton of clinical trials. So I know that I have to travel, and, you know, I'm like, oh, well, it would be cool to go to this part of the country, you know, if I have to travel. And so she knows which sites are open for which trials. And she does a great job of sort of tailoring this information to you and your needs and your wants. She's a great, great resource.
Absolutely.
I think we're going to get Katie back up here.
Yeah. I think while we're waiting for Katie, we also wanted to just mention all the different ways, you know, that these clinical trials like think about safety, and there are many different layers to it, from before the study begins, the FDA, to the, you know, the data monitoring boards, to the TDN, the Therapeutic Development Network, and all the different people involved there that review these trials and that look at the science and you know, and partner with and with the CF Foundation partners with these sponsors to bring these clinical trials, to us. So there's a lot of different ways. And then with our research coordinators, I mean, the work that they do in order to make sure that the clinical trial is running smoothly, but asking and advocating for patients, like Steph said, making sure that the trial is safe, and to do that is not cumbersome and all that stuff. It's just all of that is happening in the background.
Well, thank you very much, Steph and Dr. Uluer and Sam. This was just an amazing conversation, and I can't thank all of you enough, especially you, Steph, for just providing your experience throughout your participation in a genetic therapy trial. We are going to get to the Q&A, and so we do have a few questions that were submitted in the chat. Just a few that we have answered, but I do want to review quickly. In terms of how long the slides will be available to download, you can get back into download resources and watch replays for 12 months. And the PDF of the slides will also be available in the email you receive tonight after the event. So the first question, Dr. Uluer, I believe some of these are mostly directed towards you, but the first one is, I know about plans to use gene editing or therapy for the lungs, but will there be something for GI issues?
I mean, as far as gene therapy, certainly, especially when we're looking at gene editing, that's going to, hopefully be delivered in a way that impacts, not just the lungs, but other cells in the body. And certainly a target is going to be the GI system. There's a lot of different, challenges of delivering gene therapy to the gut. But there is right now, there's nothing on the horizon other than when we're looking at gene editing, in the future that would direct therapy there. But even then, like most of it is right now looking at how to target cells in the lung. But certainly, it's something that's being thought of. And I know Steve Friedman talked about different things that might, and Jessica talked about things that might impact the GI system as well. But right now, I'm not aware of any specific gene therapies that are targeting the gut, though.
Thank you. The next question is, you can inhale something without a vector that potentially could make a difference. Why doesn't the immune system just attack them and eat them up?
This is about the splice length oligonucleotide. So that is a really interesting question. And, when I first saw that, I was thinking the same thing. But what they do is that, like, the nucleotide or that genetic material gets broken down by certain types of enzymes, and so they will genetically or engineer that genetic material in a way that makes it less likely to be degraded in that way. It makes it a little bit more resilient. It creates like a protection for that little piece of DNA or that little strand of genetic material to get into the cell. It actually does it pretty effectively. So they're able to harness, science to make that happen.
Thank you so much. The next question, there was one in the Q&A about trials happening in Canada. I did respond to that directly, but
Okay.
there is a clinical trial finder similar to the one that we have through the CF Foundation available, and I can provide that in the chat in just one moment. The next question is, I recently saw an article about a new antiviral chewing gum. I wonder if that might be a possibility for CF patients to treat or reduce viral infections in the future.
Yeah, I'm not aware of that antiviral gum, but it makes sense that there may be something that delivers, something to where the sort of in the route that maybe viruses might use to get into your system. There's different things that we take to try to block viruses from getting in. So I can see that it's a possibility. I'm not aware of it. And I apologize.
Great. Sorry, I'm trying to add things in the chat at the same time. Okay, so the next question is, so we heard about what dosing looked like for Steph, but how might dosing differ between studies?
So was that for me or Steph?
For you, Dr. Uluer. I'm
Oh,
sorry.
wait. So can you repeat the question? Sorry, about
Yep, not a problem. So just in general, like how might dosing differ between the genetic therapy trials?
I think I had that one slide that that sort of talked about that a little bit, but with mRNA, which is not going to last very long, we're going to need to deliver that therapy repeatedly because it may only last for several hours or 12 hours. You know, we don't know exactly how long that's going to be. So that dosing and dosing frequency is something that we're working on, similar to the adeno-associated viral vectors and the HSV vectors, which each of them may last longer than some of the others, and lentivirus sort of as it may integrate may last longer. So we will try to see how often we should dose with those as we consider the immune response of the cell of the body to that vector, and as we are engineering better vectors to get around that immune response, or giving other things that might sort of tamper that response, that will determine the frequency of that. But we're hoping that some of those other viral vector treatments may be infrequent, and then the lentivirus may be a very long time, since it's going to get integrated potentially into the cell. And then the oligonucleotide, that one is not going to last very long, so that one will need to be dosed repeatedly, and hopefully has less of an immune reaction or immune response, but still has one.
Thank you. The next question is regarding vectors as well. Has any vector come to the forefront as the preferred vector for delivering gene therapy?
I mean, they all have their pros and cons. The adenovirus, associated vector, the way that it's designed can't take up the whole CFTR gene. So it is portions of it that are delivered and that will act, as well, we think, or almost as well. Lentivirus has a bigger cargo payload, so that you're going to be able to put in the whole CFTR gene, and that might be, more effective. We have done more research on the adeno-associated virus, and so there's just more experience with it. Although the other ones, there's been a lot, like for adenovirus, that is a very like a usual virus that infects the lungs. So our body may have more immune history to respond to it, where the lentivirus, which doesn't usually infect the lung, and so it's actually engineered to then infect the lung, may bypass some of that immune response. So those are some of the pros and cons. With the lentivirus, though, you're integrating, and we worry about how that then can get passed on, from cell to cell and generation to generation. So making sure that it is, that it is, modified in a way that doesn't do that. And I think that, someone just put in the chat about the negatively impacted. I mean, the 90s, when we started doing these clinical trials with the discovery of, CFTR as a gene that causes CF, we did, come across some of these immune responses, and it did lead to people having adverse reactions in other organs like the liver or other ways. So we are learning from that. So we have like 20, 30 years of experience now, and there's been successful clinical trials, and lots of other diseases also we're learning from sickle cell, from other genetically inherited diseases that have shown successes and safety. So we are now in a place that's much different than those days where we saw some of those negative responses. But we still need to be obviously super careful, and safety is number one.
Thank you. This next one is actually going to be geared towards Sam. So what are some potential barriers to participation that you have run into, or you've seen people with CF run into, and how are they handled? And that can be anywhere from reimbursements, work challenges, coordinating, with childcare, anything like that.
Yeah, we have, ways to go about distance and travel. And a lot of these sponsors for these trials contract with travel companies so that we can kind of just put the dates in and get travel booked ahead of time, so that it's not costing the person with CF and their family on the front end to book and then get reimbursed. There's no delay there. And we've been able to work around people being able to stay. A lot of our research participants have to travel as well. So just being able to set up hotels and all of those logistics. But then we do our best as research coordinators and as research teams. We really like to problem solve. So if somebody comes to us with a barrier or something we can work on, we will do our best to figure out how we can make that work alongside of the trial while getting both things satisfied. If that's pets, if it's children, if it's work, and we can maybe talk through the process of FMLA for some people if their job is open to it, or what working while you're sitting in a research visit looks like. We're always willing to try and figure it out if you're willing to be vulnerable with us and let us know kind of what the barrier is.
Perfect. Thank you so much, Sam. Just a few questions regarding timeline for drugs in the pipeline and just kind of staying up to date when it comes to new trials. You can definitely find information on the drug development pipeline or the clinical trial finder, both of which I've put in the chat. One good thing about the trial finder is that you can actually sign up for alerts. And so you can get an alert when a new trial is posted and is enrolling, or if you've participated in a trial in the past and results are available. So just a few ways to keep up to date. One other question that Dr. Uluer, I'll leave this as the last question before we close out, but just a question about how these therapies are different from a cure and if they're permanent or not.
Yeah, once again, just like modulators, this isn't a cure. It is like giving, especially with the mRNA, it's like, and like the modulators, it's like giving insulin for diabetes. You have to continue to give it in order to have that impact. So especially mRNA and other viral vector ones are just less frequent treatment, but certainly it is just providing the genetic code for the protein that is still not going to be lasting a lifetime, potentially. Some of them might be a little bit longer than others, like a lentivirus, so that might last a long time. So I think that, gene editing, where we might take out some of the material and edit it and give it back, like in sickle cell, they're able to take out those cells and replace the bone marrow with the cells that will then make the appropriate product, which is hemoglobin in that case, or a type of cell that doesn't sickle as we don't want it to in sickle cell disease. But that one is a little bit less complicated a story than with CF. So taking it out of the body and giving it back is going to be trickier. But if we're able to figure that out, and there are people who are working on this, there are people who are working on what we, again, thought was science fiction, is now a reality. And we know there are people who are taking cells out, manipulating it and giving it back to other, non-human mammals, where then those cells can be maybe taken up and, create more of a permanent treatment or potentially a cure. And that is ultimately the goal.
Thank you. Thank you, Steph. Thank you, Sam. Thank you, Dr. Uluer. And thank you again, everyone, for your participation and great questions. Before you go, please fill out the satisfaction poll, which is under the poll tab on the right-hand side of your screen, right next to the session chat. There are many wonderful resources related to this discussion, including the slides from today and the resource document found on the reception page. Please spend some time exploring the resources and download anything you want to keep. Thank you again, everyone. Have a wonderful evening.